Our speaker for today is Professor Theresa Reineke. She is currently the Prager Chair in Macromolecular Science and Distinguished McKnight University Professor in the Department of Chemistry at the University of Minnesota. She holds graduate faculty appointments in the departments of Chemical Engineering and Material Science and Pharmaceutics. She received a BS degree from University of Wisconsin, an MS degree from Arizona State University, and a PhD from the University of Michigan. She then received an NIH postdoctoral fellowship to further her research background at the California Institute of Technology prior to beginning her independent faculty career. Her research group is focused on enabling fundamental and applied technology advancements of polymers in the fields of gene therapy and genome editing, drug delivery, and sustainability. She has published over 200 peer-reviewed manuscripts and patents and manages a large group of researchers funded by several corporate, private foundation, and national funding agency grants. She has received several awards, including the 2009 National Institutes of Health Directors New Innovator Award, 2012 Outstanding New Investigator Award from the American Society of Gene and Cell Therapy, 2017 Carl S. Marble Creative Polymer Chemistry Award from the American Chemical Society Division of Polymer Chemistry, 2022 Arthur C. Scope Scholar Award from the American Chemical Society, and most recently became a fellow of both the ACS and Royal Society of Chemistry. She has co-founded 3 startup biotech companies, Tekelon Inc., Nanite Inc., and Liberate Bio. She has also served as associate editor of the ACS journal ACS Macro Letters and Chemical Science. In 2023, she became editor-in-chief of the ACS journal Bioconjugate Chemistry. With that introduction, I invite Professor Reineke for her talk today. Over to you.
I want to thank everyone and the whole team there in India, particularly Asha, for the really kind introduction today. I'm really excited to speak to you about the work that my group is doing. So I'm going to tell you about some of the work that my research group is doing and some background in it. And I want to give just a brief introduction in the goals of my total research group. So we're really interested in designing creative polymers for a variety of global applications that align with the United Nations Sustainable Development Goals. The first field here on the left is creating sustainable polymers and creating ways to make synthesis, methodology, and degradation more green and benign to the environment. The second area is essentially engineering interactions of macromolecules or polymer systems with small molecules. And this is really, exemplified by controlled release of small molecules for oral drug delivery, but we also do a lot of other delivery and fundamentals in assembly in various active ingredients and polymers that serve as active ingredients, such as surfactants. Lastly, what my talk is going to focus on is the field of designing materials, macromolecules for delivery of various biologically active macromolecules. So kind of understanding bioconjugate chemistry from a non-covalent interactive perspective, and then kind of delivery and release of these large molecules. We're interested in understanding fundamentals of synthesis, how they interact with each other, look at structure activity relationships, so really correlating performance to the fundamental structures. And then we do have a number of translational programs, and I'll just give a vignette about a couple of the companies that I've co-founded recently in the space. So I'm going to start with an introduction, and of course, we're all familiar now with the field of nucleic acid delivery because of what the field has really done for us during the COVID pandemic. So during the pandemic, we're familiar with the fact that, we had mRNA here, and we had to get mRNA into various cell types to express the SARS-CoV-2 spike protein, where our cells created an immune response against that. And in that case, it was the protein would have been the SARS-CoV-2 spike protein. But of course, really the field is primed now after FDA approval of a very inexpensive drug that was globally administered to many people for the use of this genetic material to modify various cellular protein expressions for treatment of diseases. So, the field has actually been around for probably 50 years now, so a number of decades. And researchers are looking at various nucleic modalities from various forms of DNA, whether it's single-stranded, double-stranded plasmid, various forms of RNA, such as mRNA or siRNA, to modulate expression of proteins, whether it's upregulate expression, that could be therapeutic, or even downregulate expression. Right now, this is just sort of a figure to show the pipeline of where the treatments are. And this number was quoted about a year ago, so this is probably even higher now, but 46 globally approved nucleic acid therapies, and really there's almost 3,000 treatments in the pipeline. And you can see really that cancer is the number one target that people are really developing these modalities for, but rare diseases offer a really key opportunity, and this are diseases that currently have no cure or small molecule or any option really other than donated bone marrow transplants. So this offers a lot of promise for patients who have had really zero treatment options. So, overall, this offers many modalities such as plasmids, mRNA, siRNA, things like antisense oligonucleotides that are in development by a number of pharmaceutical companies that can down-regulate expression of disease-causing proteins. And then, of course, the oncoming sort of revolution of gene editing, such as CRISPR-Cas9 and all the various editing proteins that can be used. And the promise is that these really offer the amazing possibility to repair the root cause of genetic illness by rewriting the genetic code. So this is a figure that just shows kind of the convention. So over the last 40 years, really the convention in the field has been to take a virus that we know infect our cells really well, such as adenovirus, adeno-associated virus, lentivirus. A key lentivirus or lentiviral vector that's been engineered over the years is actually the AIDS virus. And so, in fact, what they can do is take out the infective gene, engineer it to put in a corrective gene, administer this virus to humans, and that genetically engineered virus can, in fact, essentially infect the cells, but in a good way, such that it allows correction at the genetic level of this disease and modulates protein expression. So, this type of modality has really been exemplified by recent CAR T cell therapies where, we can now reprogram a patient's immune cells to attack cancer cells as an immune therapy. And one thing I just want to note is that, with the recent pandemic now behind us, we're also looking at what could be the next pandemic. And we all know that antibiotic resistance is a big problem. And this could offer, new defenses against untreatable infections where we're, instead of targeting our own cells with this type of therapy, we are enabling targeting like bacterial resistance genes or bacteria as the target to treat that as an antimicrobial. So there's a lot of wide promise to this modality. Okay, so now what is the challenge? The challenge is that DNA, RNA are really huge molecules, and when you administer them to cells, they don't just readily get internalized. And this even gets, I'm showing a cell culture here, but this gets a lot more complicated, when you're injecting material into cells. And so what we do is we need a delivery vehicle that somehow encapsulates the genetic material, that neutralizes the polyanionic charge that's available along the genetic material's backbone. So these are phosphodiester's or some modified phosphodiester polynucleotides. And so they're negatively charged, and cell surfaces are typically positively charged. They're also really susceptible to enzymatic degradation, so they're very sensitive and can degrade really rapidly, both at, on the shelf and or immediately after injection, since your body is filled with nucleases to recognize foreign genetic material trying to infect your body. So we have to design modalities that can encapsulate and protect the sensitive payload and facilitate specific interactions with different cells in an effective manner, such that when we treat specific cells, we can get the genetic material into them to perform a biological function. So another issue is that genetically engineered viruses right now, while they're effective, there's many FDA approved, and this is one example of Zolgensma, which is a drug approved for spinal muscular atrophy, and this is a genetic disease that affects pediatric patients. But you can see the price in US dollars is over $2 million per injection. So, one major downside currently of these therapies is that, the price is astronomical, and there's a lot of challenges and even questions whether or not these will ever be, reimbursed for cost of one injection by a lot of our insurance companies. And this is something that we're going through right now in the US, where some gene therapies are actually FDA approved, they're effective, but they can't really administer them to very many patients because people cannot afford, nobody really can afford that price. They also have shown in clinical trials, some other dangerous immune responses, mutagenic and oncogenic, which, can cause cancer. And then there's limitations with regard to sort of versatility, how much you can load into the capsids and whether or not these are widely available. So, what I'm going to be telling you about is my own research in the field of nucleic acid delivery. And so these offer, lower cost of production, increased stability, and high synthetic variability and increased loading capacity. So they offer a lot of versatility. And this was really exemplified again by, the fact that the COVID-19 vaccine had a non-viral carrier, which was a liposome. And, really the downside of this is that these needed to be frozen and be kept frozen in minus 80 degree freezers until basically administration. And this really prevents, administration and storage of these types of vaccines in economically depressed nations where they don't have access to specialty medical equipment like a minus 80 freezer. So my group, and what I'm going to tell you about today is our efforts to really engineer polymers, which are long chain macromolecules that can do similar function to lipids, which are much smaller molecules, more fluid and slightly less structurally stable, where these are much higher molecular weight, offer quite stable. So we typically are looking at cationic polymers, where we have like a region of amines that can then bind with DNA, and I'm showing a plasmid DNA, which is anionic. So through electrostatic interaction, which is driven entropically by the release of counterions in solution, you get a polyanion, polycation condensation reaction into these sort of colloidal nanoparticles, which are then, typically have a cationic charge and can easily interact with cell surface proteins, be internalized. We have to get these things out of the endosomes to promote facile and effective endosomal escape. If, it's an mRNA or siRNA, the cytoplasm is the final cellular destination where either genes are knocked down or upregulated, whereas plasmids and CRISPRs need to get into the nucleus to actually perform its either gene editing effect and or expression of a protein directly from a plasmid to express a proper functioning therapeutic protein. So in the field, my group really started out, and I'm showing two of my first graduate students in the field, and this was circa about 20 years ago when they originally joined my group. They're both employed by the pharmaceutical industry right now. where we took and had a hypothesis where we took and looked at the literature and found that chitosan, which is a polysaccharide, was really a non-toxic polymeric vehicle, but it had lower delivery efficiency. And then polyethyleneamine had really high delivery efficiency because the polyamine structure exhibited a cationic charge at physiological pH, which could facilitate binding to nucleic acids, but was quite toxic. So we asked the question, can we combine structural features of both a polysaccharide or polyhydroxy unit that's hydrophilic and non-ionic, as well as polyethyleneamine into polymers that exhibit both of these chemical qualities, but maybe could be fortuitous in taking advantage and be non-toxic, yet offer high nucleic acid delivery efficiency. So we originally synthesized a series of 48 structures and studied, where we looked at the stereochemistry of the hydroxy groups, the number of hydroxy groups, for example, 2, 4, and even variants that lacked hydroxy groups, and then various polyamine numbers as well as architectures. So like amines that contained where R is a hydrogen or R is a branch, so linear branch structures, as well as this varying between 1 to 6, and this is the repeat unit. So N, we also looked at, typically these polymers were shorter molecular weight or oligomeric in structure, where their degrees of polymerization or repeat unit numbers was about, on the order of 16. So a little bit moderate molecular weight. So, many papers summarized into one slide and many PhD theses. What we discovered with this series is that, chemistry and the conjugation chemistry, like binding of these two nucleic acids, significantly varied based on a lot of these chemical parameters. So the performance was quite sensitive to changing out these structural variants. And what I do also want to note are these are polyamides. And what we discovered was these polyamides degraded over time quite rapidly. So they underwent a hydrolysis reaction that was catalyzed by this sort of alpha hydroxy hydrogen bonding, activating this carbonyl for an internal nucleophilic attack by this amine. And we can also get water hydrogen bonding and hydrolysis reactions that can be facilitated to sort of chop these up very rapidly, just in a matter of about a day. You can see the degradation kinetics. Overall, the structure that had a galactose as the structure ended up being really the most, highest performing structural variant, and where this was 4, and generally the linear structure was, so galactose linear 4 was, and we called this G4 for short. And we ended up forming a small company kind of back in the early 2000s called Teculon, and they really manufactured, figured out a way to manufacture G4. They trademarked it, glyc effect, and found that it really promoted delivery of nucleic acids through caveolae mediated endocytosis, and could effectively deliver nucleic acids into the nucleus. And this just shows a picture of some in vivo data where we did direct injections into the mouse heart, and I'm showing like green glowing mouse hearts because we're delivering FITC labeled nucleic acids, and it's diffusing through the tissues. So we do get quite effective delivery performance with these in vitro and in vivo. We have further expanded sort of the structural motif, where the previous work I showed you was created those polymers via step growth polymerizations, which had limits in our hands to molecular weight. So I mentioned, we could only make oligomeric structures. Also, dispersity index can be also, limiting, where we generally, for a step growth polymerization, we're getting dispersities around 1.5 plus to 2. Whereas we asked the question, can we essentially, make similar structural variants, but use controlled polymerizations, like controlled radical polymerizations in our motifs, where, you know, we have a radical mediated polymerization of things like an acrylamide or an acrylate, but we're hanging these structural, essential structural variants off the backbone in a pendant manner. So in this example, we're hanging a trehalose, which is a disaccharide. It's very highly hydrated. And here we're modulating the charge by hanging off a primary amine, and this is called AEMA for short. And that, again, at physiological pH is protonated, and we can make these diblock structures and or statistical structures where we get this in a block, this in a statistical charge. This can then bind with negatively charged nucleic acids and make core shell structures here, showing that we can get sort of encapsulation of the nucleic acid in the center by the cationic block, but our shell now is the non-ionic hydrophilic sugar units that can facilitate colloidal stability and really sort of unique stability to lyophilization, as well as colloidal stability during injection and transfection or delivery. This just shows electron microscopy images where the scale bar is 100 nanometers, where we're taking these solutions, and as formulated, you can see the nucleic acids are about 100 nanometers. We can lyophilize them to dryness, rehydrate them, and get stable structures even after reconstitution, after physical aggregation of these. So this shows that the hydrophilic block helps with preventing colloidal aggregation. And then we can inject these into mice. And this is an injection of a nucleic acid that expresses a luciferase protein. You can see we're getting luciferase expression in the liver. So they are biologically active both in vitro, which I'm not showing in this slide, but also in vivo, which I'm showing in the slide. And this is summarized in a number of publications. Furthermore, we can kind of extend this motif where instead of trehalose, which is a non-ionic, essentially just hydrophilic, non-targeting block, whereas in this case, we're adding a targeting block. This block is non-ionic, but it's N-acetylgalactosamine, and this carbohydrate is well known to facilitate targeting to liver cells. And so this shows we have this sort of blue and red dye block, again with the primary amine, very similar architecture of a non-ionic cationic block polymer. And in this case, we've labeled the end of the block polymer with a size 7 dye. We've encapsulated a nucleic acid similarly into this core shell structure. And in this, we're just following where the polymer is depositing itself and the polyplexes in vivo by monitoring fluorescence of the actual package. The previous slide was modulating fluorescence of the therapeutic payload being expressed into a luciferase protein. So this shows that these liver, the N-acetylgalactosamine sugar targets liver hepatocytes on the asialoglycoprotein receptor, and you can see 30 minutes after injection, we're getting localization to the liver here. And then you can see this starts clearing out over the kidney, and we get actually quite high localization and lower localization to all of the other tissues within the animal. Okay, so I've kind of given you some key examples and shown you some things about how we have taken and the power of chemistry and the power of bioconjugate chemistry through non-covalent interactions and created many vehicles. And I've shown you just a few key examples of what my group has done. But what I do want to say is this recent accounts paper has really was a tour de force of my group, and one of our COVID shutdown projects where we really assessed a very comprehensive swath of polymers that have been created, and sort of published a state-of-the-art story about what's been developed, what is now clinically advancing. Again, this was published just a few years ago now in 2019. I also want to highlight, a really key article that has been a driving force for the field, and this was published by Ernst Wagner back in 2015. And so what I've shown you is that, we can modulate chemistry where you have things like within the backbone or pendant to the backbone. We can make things like homopolymer statistical alternating, or I showed some key examples of block. So I showed both alternating and block examples, but we can also modulate into many other more complex architectures such as stars, micelles, branches, and brush-based systems. And we've published in all of these areas. And then you connect these on to different nucleic acids, and they make kind of different architectures of these packages, whether they're core shell or very condensed, where there's really not a stabilizing block for colloidal stability. Next, what is also important to recognize, and this is a figure from Ernst Wagner's review paper, is that the payloads vary. So DNA, single-stranded oligonucleotides, mRNA, antisense, various protein or peptide nucleic acids, and then of course CRISPRs. So there's a lot of variability there. And when you put the variabilities in vehicles and payload together, there's really infinite possibilities. And how do we essentially, assess this field to try to pick the best possibilities that could be more rapidly clinically advanced, knowing that not one size is going to fit all for every single disease. So with this, I'm going to highlight a little bit of work that my group has been doing more recently in the field of high throughput synthesis and production in this field. So what we've done is we've taken, just linear polycations, recognizing that we can make, different distributions of the monomers in blocks. So, cation hydrophilic or, sort of statistical distribution. We can look at composition where we have like really high cationic charge or more lower cationic charge. And we can also look at variability and molecular weight. Varying sort of this hydrophilic, non-ionic, basic, or cationic balance within the polymer backbone. We've been, really inspired by a lot of work that's been done recently in the field by the Schubert, Hagenboom, Junkers, Perrier, Boyer and Cooper White, Leibfarth, and Weymouth groups, where they've done a lot of work looking at high throughput and automation in polymer synthesis, where you can create sort of structural libraries of these materials. And these groups have really exemplified a number of examples through, for example, RAFT and ATRP and many other various, polymerization mechanisms and chemistries. So in my group, I'm going to highlight just some work that has been really focused by a former postdoc, Dr. Rami Kumar, who's now at the Colorado School of Mines, and Dr. Rashad Dalal, who also just recently graduated from my group and is now in the consulting industry, where we've designed a workflow to take and make polymers in high throughput. And we've done this via batch reactors, do characterization of all their attributes such as size, pKa, binding affinity, or conjugation chemistry, along with high throughput delivery experiments in a number of different cell types, collectively taking all of this data, making heat maps, and using machine learning to not, to build algorithms to hopefully feed our algorithms, as well as identify hit polymers that we can then allow us to go back and refine our synthesis and characterization workflows to sort of design this structure activity relationships in a more intelligent manner that's allowing us eventually to predict, high performing structures for different payloads and applications. So with this, I'm going to just highlight some of the work we've done where we've taken batch reactors. We've looked at, really creating or modulating this chemistries. And, we've modulated basicity of the cation from a, more basic system to a, differing in pKa here. Again, from primary mean to different tertiary means that have this difference in basicity or pKa. Then we've modulated hydrophilic chemistries such as this MPC, which is a zwitterionic but very hydrophilic PEG, which is also very hydrophilic but slightly less than MPC, and then HEMA, which is lower hydrophilicity but still hydrophilic. And then we can make all these structural variants with different compositions. So from 100% cation to 0% cation, and this like represents, for example, like 50-50 of 50% this and 50% one of these. And then make all sorts of compositional variants. So this shows like the library of 43 polymers originally we looked at in this, where we here, you know, we're keeping the cation constant, but in these families, we're modulating the non-ionic hydrophilic group, whereas we go across the row, we're keeping the hydrophilic group constant, and we're modulating the cation here. We have also modulated, you can see within this family, P1 through 4 means we've modulated the composition. So like, for example, high cation content, low hydrophilic to high hydrophilic, low cationic composition. So this shows a lot of data, but what I can say is that, you know, we've essentially in this work tried to keep the molecular weight somewhat consistent, although there is variability that allowed us to incorporate that into our algorithm. We made all these polymers. We characterized every single one of them via NMR for the discrete composition. So what we put into the reaction doesn't necessarily mean, you know, if we put 50-50 of the monomers, we're not going to get 50-50 out because of differences in reactivity ratios. So we did actually integrate all the NMRs and assess the exact composition. We did size exclusion chromatography. We looked at dispersities, and you can see across the board our raft chemistries and high throughput yielded really low dispersities. We used titration across this with an autosampler to look at pKas of all these cations, and then electrophoresis and zeta potential to modulate it and look at like cation anionic balance, both with and without the payload. So again, taking those variants, all with similar molecular weights, and in our original work, we looked at delivery of a CRISPR-Cas9 payload, since this is really an up-and-coming modality for therapies to enable editing of the genome in a very specific manner to remediate disease. And so this shows where what we've delivered is what's called a ribonucleoprotein complex. So this is a guide RNA connected onto the Cas9 protein to form this ribonucleoprotein or RNP complex. We've added that to our polymer to create these polyplexes. With that, again, just summarizing our high throughput workload. So we took all of our libraries, made individual solutions with CRISPR-Cas9. We also did the same thing with a plasmid that upregulated green fluorescent proteins. So these are two separate delivery experiments. This Cas9, where the CRISPR is programmed to cut the genome within these cells, and the cells are engineered when they're cut. They put in an indel that upregulates M-cherry, or a red fluorescent protein variant. So we have two different readouts that allows us to assess performance and high throughput in cell culture. So again, we're looking at two payloads, plasmid and RNP. And so we looked at delivery and expression of all of our polymers with plasmid and RNP. And we did this at a variety of formulation ratios, and I'm really showing one. We did cell viability or toxicity with all of these structures. We looked at binding affinity or bioconjugation affinity of the polymer to the payload, and then assessed polyplex size and pKa and zeta potential. And so in this, I'm just showing an example in HEK cells, but we're doing this, and we have another future paper coming out, hopefully in bioconjugate chemistry within the next few weeks to show some more follow-up work on this. So what we've done is taken, like, again, the particle size, we've even looked at hydrophilicity, hydrophobicity degree, something like the Hill coefficient that we can quantitate, modulation of cation degree, and then, molecular weight, formulation ratio, percent cation and composition, zeta potential binding, affinity, pKa, etc. And what we've done is we've used machine learning as a real key tool to extract out what's really important to the delivery toxicity and uptake of a lot of these systems. So we've used both random forest classifiers as well as Shapley additive explanations. This is a machine learning algorithm based on game theory. And then we're also using various forms of neural networks now with collaborations with other computational groups. So in this work, what we did was basically, use it as a statistical analysis tool to pull out structure function trends. But right now we are doing looped learning to hopefully enable predictive, computationally predictive learning with these. So with this, what we do get are essentially structural drivers of delivery, toxicity, and internalization based on payload and RNP. And then based on polymer type and composition, on this radar plot, you can see we have all of these chemical drivers that drive performance. And essentially where, like, the arrow points, the larger the arrow, the more important this parameter is to improving efficacy for the color, which means for RNP. So for example, for efficiency, what we discovered was that the overall charge and zeta potential of the polyplexes really played a strong role in RNP delivery and high performance. Whereas this Hill coefficient also played a strong role. And this has to do with cooperativity and deprotonation of the cation. With plasmids, we found that pKa of that cation was a huge structural driver. Toxicity was really driven a lot by formulation ratio, which makes sense. The more polymer you put into the system, kind of the more toxic effects it had on the cell. And then internalization, just getting things into the cell, was actually really a variety of parameters modulated that. And a lot of this data is summarized in a couple of papers we've published. We have a new paper coming out in Chemical Science here, hopefully in just the next month. And then we have another paper coming out in Bioconjugate Chemistry that has some further follow-up work on this. But overall, after kind of showing those structural drivers, I do want to note that what we did discover was one key formulation out of this that was a sort of 50% this dip AEMA and then 50% of HIA that really allowed high performance of delivery, much higher than a lot of the commercial reagents for CRISPR-Cas9 delivery. And we verified this. This was just with our rapid kind of cell culture readout with cherry upregulation. And we've also verified this with gene sequencing and Sanger sequencing here recently. So with that, I have, really tried to push the application space in this to show that we've kind of moved the space forward, where Nanite has licensed a number of these formulations and are, really looking at translating this technology in a small startup company. And then I've also recently co-founded another company called Liberate Bio, where we're doing very similar workflows, but we're doing this with a lot of lipids and novel lipids in vivo. And so with that, what I do want to note is that, I really believe that, polymers play a role in even things like natural products, like incorporating them into chemical structures play a very high role in modulating chemistries and performance of these structures. I was going to try to tell you another story today, but I'm just going to sort of jump to sort of this second to last slide that, all sorts of chemical parameters can play a key role. And this is another example of just a vignette of work that we've recently published in PNAS and some follow work in Jack's Gold and in biomacromolecules, showing that we can incorporate things like quinine, which is another natural product. So before I was talking about sugars, here I'm actually making a natural product, cation, that can also have extremely high performance for delivery of nucleic acids into cultured cells. And the stereochemistry and regiochemistry of these monomers can play a very large role in performance, as well as composition. Lastly, I just want to put in a quick plug for the journal that I'm editor-in-chief of, Bioconjugate Chemistry. So we'd love to get your papers. We just recently published a special virtual issue highlighting a lot of researchers in India. So you can please look for that. And with that, I really, kindly thank you for all of your attention. And I do want to highlight my wonderful research group, who of course has done all of this work, and a number of very current, funding sources that we're really grateful to be working with. And with that, I'm happy to take questions.